absorption, and metabolism of folates. Folates are complex molecules that occur in a wide variety of foods of both plant and animal origin. From a chemical perspective, all folates have the same backbone, but they vary from each other mainly with respect to the presence of side group attachments to the backbone, mostly a methyl group, and the number of glutamate groups. Glutamate is one of the 20 amino acids. The majority of food folates contain multiple glutamate groups, which need to be removed to leave only one glutamate in order for folate to be absorbed. This is accomplished by the action of an enzyme that is present in the small intestine. These folates with one glutamate, called monoglutamate, are then actively absorbed by the intestinal cells. This means that there are specific transport proteins that require energy to take up these folates. Almost all monoglutamate folates are absorbed. Inside the intestinal cells, the absorbed monoglutamate folates are rapidly converted into folates containing multiple glutamate groups. This prevents the vitamin from being pumped out of the cell and thus is an important part of regulation of folate uptake. The absorbed folates can be exported from the intestinal cells towards the blood by a folate export group. Because folate pumps do not transport folates with multiple glutamate groups, these forms are retained within the cell until being shortened. Folate is transported to the tissues, including the liver, mainly as monoglutamate in the plasma, mostly loosely bound to plasma proteins like albumin. Folates are taken up in target cells by the same process by which they are taken up by the intestinal cells, which means by specific transport proteins. Folates are extensively metabolized in cells. Most important is the capacity of folates to accept or donate methyl groups. These reactions are required for many basal physiological functions, such as the conversion of amino acids, production of blood cells, regulation of the activity of genes, or synthesis of new DNA during cell division. Enzymes that catalyze these reactions are targets of several important drugs, including the cancer chemotherapeutic drug, methotrexate, which exerts its anti-tumor action by inhibiting the activity of these enzymes in tumor cells.